Welcome, everyone. Thank you for joining us for today's webinar, Validation and Implementation of Quantitative Molecular Assays. It is presented by Morgan Pence, PhD, the Director of Clinical and Molecular Microbiology at Cook Children's Medical Center in Fort Worth, Texas. My name is Judy O'Rourke, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by DiaSorin Molecular. For more information about our sponsor, visit molecular.diasorin.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Pence. I will now turn the presentation over to her.
Thank you for the introduction, and I'm excited to be here today to talk to you about validation and implementation of quantitative molecular assays. It's a bit of a dry topic, but very important for everything we do in the lab, so I'll focus a lot on the regulatory requirements for that. For my disclosures, I am receiving compensation for this presentation, and I have received research reagents from DiaSorin Molecular in the past. However, this information presented in this talk is of my own opinion and not that of DiaSorin Molecular nor of my employer, Cook Children's Medical Center. The learning objectives for today are first to define verification and validation, and then to define the verification requirements for unmodified FDA-cleared or FDA-approved tests. We'll follow that with a discussion of the validation requirements for modified FDA-cleared tests and laboratory-developed tests, and then talk about the calibration methods and standards that are required for quantitative assays. So I first want to start with kind of a definition or overview of the regulatory agencies we'll be talking about. And CLIA is one that we all hear about. It was actually developed in 1988 by the Centers for Medicare and Medicaid, or CMS. So they're really the agency that we talk about CLIA, which are the Clinical Laboratory Improvement Amendments, which define and regulate all laboratory testing, all clinical testing that's performed on humans in the U.S. And if you're interested, the actual CLIA regulations can be found in what's called the Code of Federal Regulations, which are U.S. government documents, and you can actually view all of the CLIA requirements online by Googling CFR. I also put a link in one of my last slides with resources as well in case you're interested. So another agency is CAP, or the College of American Pathologists, and they're a laboratory accreditation program. So they have a list of standards or requirements to make sure that labs are performing testing appropriately, and many of their CAP standards or the CAP standards are based off of CLIA regulations and at least meet minimum CLIA regulations and sometimes are more stringent than CLIA regulations as well. So when we talk about laboratory testing, we're focusing mostly on non-waived laboratory tests, right? So waived tests are things that are easy, have minimal harm to patients, such as things that are used in the point-of-care environment, but we're primarily focused on non-waived tests. So I want to just discuss kind of the breakdown of these are the different definitions. So FDA cleared is an FDA-approved or a test that the FDA has determined is substantially equivalent to another already approved device or test, and so this is referred to as a 510(k) approval. There's also FDA approved, so often FDA cleared and FDA approved are used interchangeably, but they actually have slightly different definitions. So FDA approved is where it has received a approval for a pre-market approval application. And this is a more rigorous application for the FDA than a 510(k). So for the most part, these are used interchangeably, and really the fine hairs of the differences don't matter so much. There's also FDA-Modified, which is where you take either of these types of tests, cleared or approved, and you modify it in the laboratory. And we'll talk about different modifications in a little bit. And then there are laboratory developed tests, which are tests that are developed wholly or in part by the performing laboratory. And this may include analyte-specific reagents, or ASRs, which are reagents such as primers and probe sets that are sold by manufacturers and used to develop a test in the lab. So historically, a lot of labs would design their own primers and probes, but as technology has advanced, a lot of these can be purchased from manufacturers. And in regards to validation requirements for what we're concerned about in the lab with bringing in new tests, FDA-modified tests and LDTs are considered the same. So I've had people tell me or try and argue in the past that because the test was FDA cleared, it's okay, they don't have to do as stringent of a validation as a laboratory test, laboratory developed test. But actually, as soon as you modify it, it becomes off-label and treated like an LDT, and we will talk about that as well. So let's define verification and validation. And this is actually a little bit confusing because different groups have different definitions. So as defined by CLIA or the government, verification is a one-time process prior to implementation of the assay where you're demonstrating that that test performs as expected. And their definition of validation is an ongoing process to confirm that the test continues to perform as expected. So I would say most people do not use these definitions, but instead more refer to the CAP definitions. So CAP defines verification as the process by which a lab determines that an unmodified FDA-cleared test performs according to the manufacturer's package insert. And validation is basically the same thing, except you're showing that a laboratory developed test or a modified FDA test approves or performs as it should. And so these are used often interchangeably. They're pretty close in meaning, but verification is basically when you're bringing in an unmodified FDA cleared test, and validation is if you're bringing in a laboratory developed test or a modified FDA cleared test. So which one is correct? Validation and verification are used interchangeably, and primarily when laboratorians are talking about these processes, they're talking about the initial process that is done when the test is brought in to ensure that it performs as expected. And what CLIA defines as validation is primarily what laboratorians refer to as quality assurance, which is once the laboratory has validated or verified an assay, we then make sure that the test performs as expected as we continue to run it and perform patient testing. So quality assurance measures include quality control, proficiency testing, calibration verification, and analytical measurement range verification. And we'll talk a little bit more about those towards the end of the talk. So as far as what's required when it comes to verification validation, and we're going to focus when talking about verification validation on the CAP definitions. So laboratories are required to perform this initial verification or validation before the test is used in patient testing. So you can't, bring it in and start testing patients and then ensure that it's performing well. And this is required for any non-waived test, a new test method, or a new instrument. So even if you run a CMV quant, if you bring in a new platform, you have to validate that. And CAP specifically states that not only does this include instruments of the same make and model, but also loaner instruments. So if something happens to your instrument and the manufacturer gives you a loaner instrument, you actually have to validate that as well. And there's no exception for tests introduced prior to a specific date. So if you have a test that was introduced, back in maybe the early 2000s, you still have to have verification or validation records for that as well. In regards to FDA-modified test, what is it that constitutes a modification? And basically, it's anything or any modification that's made to the package insert. And these include things such as specimen source. So if the test is only FDA cleared for CSF, but you want to validate it in-house for blood and BAL to test patient specimens, now that's FDA modified or off-label, and it's treated as a lab-developed test. This also includes things like pooling specimens, diluting specimens, changing the collection device. So if it's approved for universal transport media and you want to use M4, which is a different type of viral transport media, that's off-label. patient population. So there's some tests out there that are not FDA cleared for patients under 14, for example. So if you test that patient population, it's off-label. And also instrument changes. So if you have a quantitative molecular assay that is FDA cleared for use, for example, with the Roche MagnaPure instrument, but you want to use the bioMérieux EasyMag, that's off-label as well. And as I mentioned a little bit ago, FDA-modified tests are considered the same as LDTs when we're talking about laboratory regulatory standards. And once you modify a test, the reason why it becomes similar to an LDT is because it's now outside of the FDA-cleared scope. So when companies submit assays to the FDA for clearance, they're very defined and have very defined criteria and specifications. But once you change a specimen source or pooling, now you've changed the intent and the scope of that assay. So it is more similar to a laboratory-developed test. And in addition, once you modify it, any test, it becomes high complexity. So if you are using an FDA-cleared test that's considered moderate complexity and you're using it off-label, it's now automatically high complexity, which changes the personnel requirements as well. So before you start your verification validation process, you want to determine what specimen types will be included, right? So are you going to use it off-label? Are you going to stick to the package insert or whatnot? And specifically, when you're thinking about blood, you can't be so non-specific. So, right, we think about quantitative molecular assays, and blood is one of the main specimen type that they're used for, but you really have to define this as, is it whole blood, is it plasma, or is it serum? And then if you're going to validate plasma or verify plasma, you must define which type of plasma, right? So we generally in microbiology or molecular microbiology think of purple tops with EDTA plasma, but there's also different types as well, such as those with acid citrate dextrose and also lithium and sodium heparin, but those are not used typically for PCR because of the inhibitory effects of the heparin. It's important to note what you're validating as well, because viral loads may be significantly different between blood sources. So for viral loads for things like CMV and EBV, we see significant differences between whole blood and plasma. So you want to make sure you know what you're validating and stick with that. The specific requirements for verification validation are listed here, and these are pulled from the CAP checklist. So if you are verifying an FDA-approved non-modified test, so you haven't changed anything in the package insert, you have to include accuracy, precision, reportable range, reference range, and analytical sensitivity or limit of detection for that assay. And we'll go through these each in more detail. Now, as far as defined actually by CLIA, you don't have to do analytical sensitivity, but CAP has said that for quantitative tests that you actually do have to do the limit of detection. So that is an extra requirement for CAP. For FDA-modified or lab-developed tests, you have to do the same thing as the FDA-approved non-modified tests, but also test analytical specificity or interferences. And the CAP standards that refer to each of these are listed in the column on the right. So if you want to refer to the different checklists, these are from the All Common checklist and the microbiology checklist if you want more details as well. In addition to those specific requirements, there are also a couple of more requirements. So one is that the instrument must be verified or validated in the location in which patient testing will be performed. So if it's moved, if you validate it in one place and move it somewhere else, you have to determine if that will affect the performance of the assay. And so CAP is a little bit gray on this, and they don't give definitions of, if it's moved over 25 feet to a new building. They don't specifically say, but the lab is responsible for determining if they move it, if it may affect the performance of the assay. And if it does, then you'll have to re-verify or revalidate. And so as an example for this, we actually moved labs a new a new tower in our hospital was built a couple of years ago, and we moved, and it was a completely different kind of building or section in the hospital, and so we revalidated, re-verified all of our assays. For each verification or validation that's performed in the lab, there must be an evaluation that's signed by the laboratory director prior to use. And so what this is a summary. We also, in our lab, put all of the data with that as well, and a binder, and then that must be retained as long as the test is in use and for at least 2 years after a test is discontinued. So make sure you have all of those documented, and then some CAP inspectors come in and like to look at the validation verification summaries. So those are the general kind of requirements and overview as far as verification validation. But let's move into more specifically discussing verification validation of quantitative molecular assays. So step one is calibration. Once you've decided, this assay and the platform that you're going to use, you want to do calibration. So there should be an image here. I'm not sure if it's showing up. But this calibration is establishment under certain conditions between the instrument response and the corresponding concentration of an analyte. So if your analyte is 500, is that what is being reported by the instrument? And the lab must have defined procedures for calibration. And so there's our curve, okay. And it doesn't necessarily say, CAP doesn't define what that process has to be, but just instead says that the lab has to have specific procedures that define how they're going to perform calibration. And on the right here is a calibration curve shown from one that we've run for one of our EBV quant tests. When you're deciding, when you're getting ready to do your calibration curve or your standard curve, there is a decision about what materials to use. So if you're using an FDA-approved or cleared assay, the manufacturer should have instruction for those on how you calibrate it and what specific materials you use. And often those are provided by the manufacturer. For FDA-modified tests or lab-developed tests, especially those using ASRs, you'll want to purchase materials as well. So you want high-quality materials that have test system and matrix appropriate target values. And matrix is referring to your specific source, so plasma, CSF, etc. So calibration materials are available from the manufacturer, often for FDA-approved tests, or third-party vendors as well. And I want to make the point that all calibration materials are not equivalent. So, there's lots of CMB calibration panels out there, for example, but the reliability and repeatability is not always consistent. So there are some manufacturers that have more robust calibration panels than others. And it's important to use high-quality calibration materials that have less variability so that your assay is consistent and you don't run into any QC failures or any issues with variation in your assay reporting. Calibration materials should contain whole virus in matrix-appropriate materials. And so this is to be the most closely related to a patient specimen as possible, so you have it mimicking your human specimens. So some calibration panels are true EDTA plasma, where others are protein matrices that mimic a specimen. So that's something to keep in mind. And if you are using one that has protein matrices, does it adequately mimic it? Does it perform well? So something to keep in mind when comparing that. And you could also talk to your colleagues, your clinical colleagues, for recommendations on which manufacturers provide high quality materials, or try them yourself, or there are some references in the literature looking at performance and comparing these against one another. So now that you have materials picked out, you want to know, does your platform allow for a stored standard curve? So can you make a calibration or put together a calibration curve and then store it and continually use it each day? If not, and some instrument platforms require you to perform a calibration each day. And so then you'll need to just create a new standard curve each day of testing. When you're creating your standard curve, you'll want to run your calibrators anywhere in duplicate to sextuplicate, which is 6 replicates. Some FDA-approved tests require these only to be run in singlet, but a lot of times, we'll run ours if we're creating a new calibration curve in sextuplicate, just to make sure we have, a lot of replicates to make sure we have an accurate curve and detect the variability that may be there. And you may want to consider running more replicates for the lowest or the lowest 2 calibrators, because in general we see more variation at the lower end of the curve, and that can help you kind of detect the greater variability there. So sextuplicate, sometimes potentially even more. Once you have then run your calibrators and created your standard curve, you'll want to perform what's called regression analysis. And this may be performed automatically by the test system for FDA-approved or cleared tests and may say this meets the requirements and accept it. Otherwise, you'll likely have to analyze it yourself and determine if it is a good fit or not. Is it a good curve to use? And if not, you'll want to then recalibrate. So in regards to what to analyze, there's a few different things you want to look at, and this is one of our curves here. This is actually a really beautiful curve. They don't always look this nice, but it is a nice example. So if you remember, I don't remember if it was grade school or middle school, but we learned about y equals mx plus b, which is the equation for a line, and m equals your slope, and b equals your bias, and then r squared is another point that will be analyzed, and this is your coefficient of determination, which will assess the fit of the curve. So your r squared should be as close to 1 as possible, or 100%. So if you can see at the top of the graph on the right, the r squared is 0.99, so 99%. So that's a very good fit. So some people will, think, oh, you just look at the r squared and you're done if you have a good r squared, but that's not true. Sometimes you can get a good r squared, but still not a very good curve. So in addition, you want to look at slope, so that m, and it should be close to 1. So in this case, it's 1.004, so that's a very good slope. And then your B point, which is your bias, should be close to 0. So there are no definitions of, this is what is an acceptable R squared, slope, or bias. You just want to make sure that they're as close to 1, 1, and 0 respectively as possible. And if you have a high R squared, but your slope is not close to 1, or your bias is really high, 0.8 or 1, then you'll want to run a new curve. And this is something you'll kind of develop a feel for after a while, but there are no defined criteria for what makes it acceptable. In addition, you'll want to look at the coefficient of variation, which is a measure of relative variability. So we use the DSOR and molecular quantitative assays. This is one from one of our assays, I believe EBV, and this is analysis from the instrument. But if you look at the coefficient of variation on the right side, so in the far right column, if you look for your higher calibrators, your mid to high calibrators, your CV should be low, less than 10 to 15% ideally. And at your lower calibrators, you're going to have more variance, so your CV is automatically going to be higher. And it's recommended to keep that within 20 to 50% or so. So if you see that start to creep up in 60 to 70% or higher, you're going to run into some issues there. And that's really just to make sure that there's not a lot of variation in your curve and within your assay. Once you have your curve figured out, then you'll want to start running patient specimens. And with patient specimens, you'll want to run quality control. And the quality control requirements also apply to patient testing after you implement it. And so as far as what's required for daily quality control is for quantitative assays, it must consist of at least 3 controls. So a low positive, a high positive, and a negative. And similar with a lot of these things, there's no definition of what constitutes a low or high positive. It's kind of a relative term. CAP does recommend that if there is a specific cutoff value that you're using for your assay, that you use a control near that. But for a lot of these quantitative assays, there's not a defined cutoff. So for CMV quant, for example, there's not necessarily an amount that determines disease versus not disease. So just generally a low, high, and negative. Controls must assess the adequacy of the extraction amplification. So it's important to treat your controls as a patient specimen. So they should go through the entire process through extraction and amplification to control for all steps of the test. When you start your validation or verification, you'll want to start with accuracy. So accuracy is comparison of your results by your new method to what their established results were. So you can either use a reference method, but a lot of times we don't have the gold standard method available, and we're often comparing just to another established comparative method. So an assay by another manufacturer perhaps, or from another lab. And so you're looking at how close a test result is to the analyte's true value. And you want to use matrix appropriate materials, and so preferably for accuracy studies, patient specimens are best. That may not always be possible depending on what the assay is and depending what your lab has available, but you can also use matrix appropriate reference materials or other commutable materials. However, CAP defines specifically that you can't use routine quality control material or calibrators that are used to calibrate the test. So primarily patient specimens, but you can use reference materials as well if necessary. And we'll talk about places to get some of those in just a moment. So the big question with accuracy always is, how many samples do I have to run? What is required? What's the minimum I can get by with? And some of the standards say kind of more general requirements. So one of the micro standards on the CAP checklist says an adequate number of positive and negative specimens representing the sample types used in the assay. So that's not the most helpful. But they do say that specimens may be spiked if patient specimens aren't available or you don't have them spanning the entire range. So more specifically, CAP does have a couple of checklist items that refer to accuracy for FDA approved and then FDA modified and LDTs. And for FDA approved, it's kind of like the general guidelines. They don't give a lot of guidance. So they simply say that the laboratory verifies or establishes accuracy using a sufficient number of characterized samples. And again, that routine quality materials are cannot be used, but they don't state how many. So generally, even for our FDA-approved tests, we use the guidance from FDA-modified or LDTs. So they do give specific numbers for that, and so for qualitative tests, it's a minimum of 20 samples, and this includes positive, negative, and low positives. And when you're talking about quantitative, we'll talk about quantitative in a minute, but this includes positive, negative, and low positive. Specifically for quantitative tests, the number is the same. It's a minimum of 20 samples, but they say that it must span the analytical measurement range, which is basically the range of the assay. So if your assay goes from 500 to 5 million, you want to have samples that fall across that curve and not just all of them falling around 50,000, for example. And you can use mixtures of samples if you need to get some of the concentrations that you need. So I will add that CAP doesn't specifically say this, but this minimum of 20 samples is referring really to the sample type. So if you're running a quantitative molecular assay and you decide you want to do plasma and BAL and CSF, for example, and if you want to run them all, then you really need to do 20 samples per specimen type, so 20 plasma, 20 CSF, and whatnot. So they don't specifically state that, but whenever I've asked them, they do say that it really means by sample type because they're all kind of little mini validations, or you're validating each source. So that's something to keep in mind. If you don't have 20 samples, and for things with assays that have less common analytes, you can use fewer than that. If you do, the laboratory director must write down, and it must be in writing, the criteria that are used to determine the appropriateness. So if you only have 10, the lab director has to kind of sign off why that's acceptable and why you couldn't get any more samples. So CAP put these requirements as far as the 20 specimens. That was added to their checklist in July of 2016. Prior to that, they didn't state specifically how many samples needed to be used. So if you validated an assay before that CAP standard came out, you still need some records of performance, but they do say that you can use either proficiency testing results, alternative PT, swapping samples between labs, or quality control to show that you may have not had that many originally, but that your assay is performing as expected. I would argue in a lot of cases that you shouldn't have less than 20 samples for a given analyte because there are a lot of ways that you can acquire samples. And so, one of the most common ways is if you're simply just switching platforms. You're going from one CMV quantitative assay to another, and you are likely going to use your samples that were tested on that previous method. You can also share samples between labs or between colleagues. So I frequently, if I want to validate something we don't have, or it's a new transport media, et cetera, I'll shoot my colleagues an email and ask if they have specimens they can share. You can also use CAP proficiency samples. Some of the reference labs will actually sell samples, so once they're done past their storage date, you can purchase them and they will give you the values to do that. So we've done that in the past. And then you can also spike samples as well. So I would not recommend doing your entire validation verification using spike samples because it doesn't as closely mimic human specimens, but a combination of these can be used. In addition to accuracy, one of the requirements for verification validation is precision or reproducibility, and that's looking at if you take the same sample and repeat it multiple times, does it still give the same result? And there's two types of precision. There's within-run or intra-assay precision, which is where you want to test the specimen in multiples on one run. So, at one point, instead of running it in singlet, run it in duplicate, triplicate, etc. And then there's also inter-assay precision, which is between run. And so you want to take a subset of those specimens. And I should make a point of this. You might as well kill two birds with one stone. If you're already using these samples for accuracy, you don't need separate samples for precision. You can use your accuracy samples for precision. So for inter-assay or between-run precision, you want to test it on at least another day by a different technologist. And so you want to do this also for each sample type that you are validating or verifying. So run it one day, wait a day or two, have another tech run it, maybe do that a few times. There's no defined numbers for precision, so there's nothing saying, oh, you have to do 3, you have to do 20. It's basically up to you as a lab what you want to do and how many you want to run for precision. Another requirement for validation verification is analytical sensitivity or limit of detection. And so per the all common checklist, LOD studies aren't necessary for FDA approved or modified tests. I'm sorry, that's a typo. That should be FDA approved or cleared tests. But per the microbiology checklist, the LOD must be verified or validated for all quantitative tests. So even though we got away with it in the all common, the microbiology checklist says that we have to do it. And so limit of detection studies, the limit of detection is defined as greater than or equal to 95% of replicates are detected. So typically 20 specimens are used, and as long as 19 of these are detected, you can say that that is the limit of detection. However, if you run 20 samples and more than one isn't detected, you'll have to increase your number or increase your LOD. Some people will do less if they're running an FDA-approved test. If they're staying on label, they may just run a few replicates to verify the manufacturer's limit of detection and do a more extensive limit of detection study if it's off-label or an LDT. Analytical specificity or interference testing is only required for FDA-modified and LDTs. So if you're using FDA approved and you're sticking to everything in the package insert, you do not have to perform analytical specificity. That's actually already reported in the package insert. And what we do in those cases is we will actually just copy the interferences that were tested if we're staying on label and put that in our verification summary. When you do this testing, so the point of analytical specificity or interference testing is to ensure that related analytes or foreign substances don't interfere. So for example, if I keep going back to CMV quants, but primarily that's because that's the most widely used quantitative assay. But if you're running it, you decide to run it on BALs, you want to make sure that it doesn't interact or normal respiratory flora don't interfere with the assay. And so you can perform these studies yourself. You can spike high concentrations of organisms into the specimen and run it and make sure it doesn't interfere. Or if there are published references, CAP says you can cite those. And if you do find any interferences, a lot of times there's not, but if you do, you have to document that and have a plan of action when they are present. The reportable range is also required for both FDA-approved and FDA-modified LDTs. And this is the range of values that can be adequately or accurately measured by the test. And so when you're using your calibrators, that's really going to help guide or establish your reportable range. You want to stay within kind of the highs and lows of your calibrators. So one of the panels that we use for our calibrator panels, the lowest calibrator is 500 IUs per mil or 2.7 logs, and the highest is 5 million. So our range for that assay is 500 to 5 million. We found that it performs accurately along that specific range. And you can put this either, you have to have it documented somewhere, so either in your validation or verification in your standard operating procedure, or some people also will put the range on their report as well that goes out into the electronic medical record. You cannot report outside the range of calibrators unless you validated that. So if you want to report, so for example, on ours, if we would get, in the example I mentioned above, something that was greater than 5 million, we simply report that as greater than 5 million. You can do further validation studies to dilute a specimen. So if it would come back, to dilute it and be able to give it a specific value, that does require further validation, but that does allow you to report outside of the range of those calibrators. Once you have it validated, you also want to think about how you're going to report your quantitative assay. And one of the big questions that really there's not a good answer to is, should quantitative results be reported as integers or log values? So integers is just something, like 321, or do you want to put it and simplify it into log values? So there's arguments on both sides of the situation here. So integers are easier for non-laboratorians to understand. I found that non-laboratorians have a difficult time grasping what exactly a log value is. However, integer values can cause overinterpretation, and I've seen this on multiple occasions. And so an example of this would be if you have a quantitative assay and you report, 100,000 copies per mil, IU per mil, whatever, and then maybe they run it again the following week, and now it's 200,000. And so that looks really different. It looks like you've doubled your viral load, but depending on the specific assay, that may actually be within the variability or the precision of the assay. It may actually be statistically equivalent and not different. But the physician will likely look at that and think that the viral load is increasing. And I've given those logs and values to where yes, it looks like it's going from 5.0 to 5.3, but you could potentially give them more guidance on how to interpret a log value. Log values in general allow providers to more easily track trends. So although they often have a little bit of trouble understanding what exactly a log value is, it is easier for them to look at and track. And so I've just given an example here of two reports of 5.12 and 6.34, and you can clearly tell the difference there and could easily track it as sequential viral loads are obtained. There are some integer values that I put here, and I put it without commas because one of our, the previous EMR that we used wasn't able to put commas in, and so potentially at first glance, you may not interpret these correctly. It's 131,000 and then 2.1 million, but you may confuse those, or the physician interpreting the results may confuse those because they're not easily interpretable. So I should go back. There's not really a defined better or worse, and a lot of labs will report both the integer value and the log value. We have switched over to reporting only the log value, which we did get a little pushback at first, but we provided the physicians the information with how much variability there was in our assay, so look for this much for it to be considered a true change. And they've since kind of adjusted to that. So, but that's something to think about before implementing it, and how physicians look at it and how they interpret it. And there's no wrong or right answer. I think you could argue either, but you want to look at what's most important or what's most helpful for your institution. Another question that you will get is what threshold or cutoff defines disease? And this is something clinicians often want answered, but we often don't have an answer to. And part of that is due to the fact that viral loads vary within labs and between labs. And so a single assay within a lab may vary up to a half a log or more potentially, and may vary more than one log between labs. So that's tenfold difference. So it's hard to define a cutoff or a threshold value when some of the viral loads vary so significantly. The threshold that defines disease varies by assay, and it often can vary by patient. So some exceptions are things like HIV and the hepatitis, because if you have any amount of HIV, you have HIV disease. But for other things, such as thinking about Epstein-Barr virus and post-transplant lymphoproliferative disorder, there's no definition or no cutoff of this amount is what determines if the patient has PTLD. So there's studies on that showing that maybe for some patients it's a few hundred, maybe for some it's closer to 10,000, but you will get questions, and that's something that you can't really answer. Viral loads have also been shown to be linked to drug regimens used for immunosuppression. So depending on what chemotherapy a hem-onc patient might be on, for example, or if they change the drug regimen, it also can change the viral load. So that may be actually affecting what is changing rather than the viral load changing on its own. In general, we don't know what the cutoff or the threshold is. And again, our clinical colleagues want kind of a hard number of, this is what we're looking for. But for a lot of these assays, we can't give them an answer. It's more about replicate testing, repeat testing, and tracking the trends instead. So once your assay is implemented, you'll want to think about quality assurance. So this is what CLIA refers to as validation or ongoing assurance to make sure that your assay is still performing as expected. So now you've implemented an assay, and now what do you do? So there's different definitions for calibration, and we'll talk about those. So we've talked about calibration or creating your standard curve, but if you have one of the platforms that allows you to store your standard curve. So I mentioned we use the DSR molecular liaison or instrument, and we can store our calibration curves for a long time. So what you'll then be required to do if you're able to store your standard curve is perform what's called a calibration verification. And this is just showing that, if you've had that curve for months, that it's still performing as expected. And so then we'll talk about the intervals for this, but basically confirming that your calibration settings are still valid over time. There's also the analytical measurement range, or AMR, and this is the range of analyte values that a method can measure directly without dilution or concentration. So as the example I gave earlier, the assay going from 500 to 5 million IUs per mil, that would be the analytical measurement range. And so one of the QA requirements is to perform AMR verification, which is the process of confirming that your range remains valid. And then there's recalibration, which is basically where you're creating a new standard curve, so following that initial calibration procedure we talked about. And this is performed when either your calibration verification or your AMR verification fails. Again, these, if you have an instrument where you have to create a standard curve each day of testing, these QA measures are not going to apply to you. For calibration verification, acceptable materials must be matrix appropriate and include, you can actually use the calibrators that you use to make your initial standard curve by running them as unknowns. You can use preferably a different lot number, but that doesn't always necessarily have to be the case. Materials provided by the vendor for calibration verification. You can use previously tested patient specimens that you know the values of. Primary or secondary standards or reference materials that are matrix appropriate, so again, mimicking patient specimens, or proficiency testing material. It's specifically defined by CAP that you cannot use routine controls for calibration verification. And as with a lot of the things that I've been mentioning in this presentation, each lab has to define the limits for accepting or rejecting the calibration verification. So, you run your calibration verification and you look at the variation between your results and what the expected results are, but there's nobody out there defining what that is. For some of the FDA-approved assays, they may have that. But for the most part, like when we're thinking about ASRs, like with the DSR and molecular instruments, those we have to define the calibration verification requirements or thresholds ourselves. And so that's put in a procedure. And if the calibration verification fails, then you must recalibrate your curve. AMR verification may actually not be required depending on how you're running your specific test. So if when you run your calibration verification, you're using low, midpoint, and high values near the stated AMR, so near the high and low, and if your calibration verification meet your acceptance criteria, then you actually don't have to perform a second verification. You don't have to do a specific AMR verification. That's already been met by your successful calibration verification. However, if your calibration verification materials do not span the full AMR or the lab extends it beyond the manufacturer's recommendation. So, for example, if the high end of our AMR is 5 million and somebody has validated dilution beyond that, then you would have to do a separate AMR verification to show that you get adequate or accurate results across the entire range of the AMR. But really for most labs, a specific AMR verification won't be required because it will be covered by a calibration verification. Acceptable materials for AMR verification include, again, they should be matrix appropriate, but linearity material. So CAP does have a linearity panel for some of the viral load tests that you can use. Proficiency testing material, previously tested patients, primary or secondary standards, calibrators, or basically the difference between this and calibration verification is that you can also use control materials if they adequately span the AMR. And as with calibration verification, each lab has to define the limits for accepting or rejecting AMR verification results. So for example, you may, during validation, realize that the variation in your test is, let's say, 0.3 log. So it might be that when you're doing your calibration verification, AMR verification, the unknowns have to fall within 0.3 log of your knowns. So something like that, but you do have to define the actual limits. As far as the interval or how often you have to do this, calibration verification and AMR verification must be performed at least once every 6 months. So if you are allowed, if you can on your instrument to store a standard curve, you can store it for up to 6 months or longer if your calibration verification is successful. You also must perform calibration verification and potentially AMR verification when there's any changes in major system components. So if you have an engineer come in and replace a part, for example, and also at reagent lot changes. So what we try to do is order reagents, enough reagents to get us to 6 months. If you order your reagents more frequently, every month or so, you're going to have to do calibration verification as often as the reagents show up. So we try and order about 6 months of reagents at a time so that we're only doing calibration verification around the 6-month requirements. And again, the acceptability criteria must be established by the lab. In addition to calibration verification, AMR verification, one of the requirements as well for ongoing quality assurance is what's called comparability of instruments. So if the lab uses more than one instrument or method to test for a given analyte, those have to be compared against each other at least twice a year. So this could be, for example, let's just say CMV Quant. If you have 2 instruments that run it, same manufacturer, same platform, those have to be compared. If for whatever reason you run CMV Quants on 2 different and 2 instruments by different manufacturers, those have to be compared as well. As far as what you can use for comparability studies, if you are only testing on the same instrument platform, so for example, if you use the DSOR molecular liaison instrument and you have 2 of them, 3 of them, but it's all on the same instrument platform, you can actually use quality control data as your comparability data. As long as the reagents are of the same manufacturer and lot number, you actually can just use the quality control data from your daily runs to compare and show that the instruments are comparable. You also, if it's the same platform or if you have different platforms, you can also use human specimens. These are preferred over stabilized controls so that it's more similar to kind of it's the same as your human samples and avoid any potential matrix effects. So when we do comparability of instruments, we use primarily human samples. As with many of the other things in this talk, the acceptability criteria are not defined by regulations, nor are the amount of samples that you have to test or how many quality control data points. So nobody has put a definition on this. CAP has not said, that you need a specific number of QC points or that you need a specific number of human samples. So that is up to you in the lab, but you do have to define it and have that in a procedure defining what you're going to run and how many of what you're going to run. So we've covered a lot today in this talk, and the basic conclusion is that there's a lot of regulatory and standards requirements as far as verification and validation and quality assurance in the lab. So it gets very complex. It often gets, causes confusion. I've gotten a lot of questions from colleagues and lab techs about, why do we have to do this? Where is this defined? What do we do with this? And they are complex. But don't be afraid. Consult your CAP checklist frequently. They are very well defined there, and with time, CAP has gotten very or more specific with their requirements and more defined, so it's easier to understand. And don't be afraid to consult colleagues who have experience and ask them if you do have questions. And I put an important point here that this includes chemists. So we don't often think of consulting with chemists in the microbiology lab, but they validate and verify quantitative tests all the time. Essentially, most of their tests are quantitative, so they are a great resource. If you have a chemist in your lab, even you can ask questions there. If you make sure that your lab has all of the following, you will be just fine and not have to worry about it. So even though it's complex, there's a few things that you'll need. So if you have your verification validation summaries signed by the medical director with supporting data, you'll want to have that and make sure you keep those for at least 2 years after the assay is finished or not being used. You want to make sure you have test procedures for all of your specific assays that you're running. Make sure you have reliable calibration materials to minimize variation and minimize QC failures. And make sure that you have a procedure either in your specific test procedure or have a specific procedure for how you're going to perform calibration verification, AMR verification if necessary, and recalibration. Make sure you have, what samples or what calibration panel you're using, what the acceptability criteria are for that, and what you're going to do if it fails. That's one of the big things, is that CAP doesn't specify, the acceptability criteria, but you must have them and you must develop them as a lab. So if you have all that, you'll be just fine. It's not as hard as it seems. Once you get going, it gets a little easier. I've included some references and resources here if you have further questions. So the CAP checklist, the ones that I have included the numbers from are the 2018 versions. So there's the All Common, which are the COM standards, and the MIC standards are from the microbiology checklist. I've also included a link if you're interested in going to the CLIA requirements and the Code of Federal Regulations. You can read through those at your leisure if you're interested. And then don't forget that you can reach out to colleagues. There is the AMP listserv, there's the Division C listserv in microbiology. You can email other friends and colleagues. And don't forget that you can ask your chemists as well. And with that, I'd be happy to take any questions. Thank you.
Thank you, Dr. Pence, for that informative presentation. I would also like to thank DSOR and Molecular for making today's educational webcast possible. As a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Goodbye.